Hey there, farmers. Ever wondered what makes soil truly healthy? Sure, you might think of nutrients, water, and organic matter, but there's an entire hidden world beneath our feet that's just as crucial. Yep, I'm talking about soil fungi. Today, thanks to our friends at microBIOMETER, we're diving into this fascinating underground ecosystem. We're going to explore two key groups of fungi: arbuscular mycorrhizal fungi, or AMF, and saprophytic fungi. So, what's the big deal about AMF? These fungi form amazing partnerships with plant roots. They help plants absorb nutrients and water more efficiently, boosting growth and resilience. Imagine having a secret ally underground, working tirelessly to keep your crops thriving. Now let's not forget about our decomposers, the saprophytic fungi. These guys break down organic material, returning vital nutrients to the soil and enriching it. They're like nature's recyclers, making sure nothing goes to waste. But how do you measure and monitor these important fungi? That's where the microbiometer comes in. This handy tool lets you get a snapshot of your soil's microbial community in just minutes. No more waiting weeks for lab results, just quick, actionable insights right at your fingertips. Understanding the balance of fungi in your soil can revolutionize the way you farm. It can improve plant growth, boost resilience, and maximize productivity. Who wouldn't want that? So let's get ready to uncover the incredible world of soil fungi and see how they can contribute to the success of our farms, gardens, and landscapes. Let's see what Laura Decker, the president of microbiometer, has to say.
Hi, I'm Laura Decker, the president of microbiometer. This video is just to tell you a little bit about microbiometer so you can get the most out of your product. The microbiometer was designed by Dr. Judith Fitzpatrick, who was an immunologist who was fairly obsessed with microbes. She wanted to give growers more information about their soil's biology, and since she knew they were living organisms, she wanted a test that was quick and easy and accessible and affordable. The result was the microbiometer. Here it is as a kit. It includes everything you need to test soil and return soil microbial biomass and fungal to bacteria ratio. We use a cell phone complete with instructions in 18 languages and videos to make sure you can get the most out of your test. The test is best used on agricultural soil as growers transition from more conventional practices to more regenerative practices and try to harness the incredible power of soil microbiology to improve plant health, plant yield, and reduce potentially harmful and expensive inputs like fertilizers, pesticides, and herbicides. The test is best used on field moist, fresh soil that you collect and test as soon as possible. You want to make sure that your soil is not overheated or frozen or dried out between when you collect it in the field and when you test it, either at home, in the field, or in your office. After you take the test and get the results, what you're going to do is think about comparing either over time or over space. If you're doing space, what you want to do is take samples from fields around the rhizosphere. Biology is the most active around active plant roots. So you want to go to, say, 2 fields that you're doing different practices on, perhaps different cover crops. Take a sample on the same day and test it on the same day to see perhaps which cover crop is growing and harboring the most microbiology in your soil. You also might want to test over time to see if a particular practice that you're trying out or using is actually growing your soil biology or changing your fungal to bacteria ratio. Many growers, as they seek to improve soil structure, want higher fungal content because fungal content is the best grower of soil structure. So if you're doing over time, what you want to do is take soil samples from approximately the same place, again around a growing root if it's there, and be able to assess what was the impact to your practice or your input on the soil biology. The more you test with the microbiometer, the better a sense you'll get of both your soil's natural microbial patterns and also how your growing and soil stewardship practices are impacting soil biology. We really hope that you enjoy the microbiometer and that it helps you become a more responsible and efficient growth.
The microbiometer estimates the microbial biomass in a sample of soil and now can tell you the ratio of fungal to bacterial biomass. Bacteria, some of them can fix nitrogen, they break down organic matter, and they form the base of the soil food web. Fungi create soil structure through secretions and cross-linking. They're critical for nutrient cycling that can improve plant health and cause enhanced growth, and their lower turnover relative to bacteria means they are important for carbon sequestration. We can further categorize fungi into two types, saprophytic and mycorrhizal. Saprophytic live on dead material, whereas mycorrhizal live close to or in the root and are symbiotic with the plant. Saprophytic are present in all soils, whereas mycorrhizal are associated with 90% of all plant life. Saprophytic function to recycle, especially hard to digest foods like wood, and then they release these nutrients to feed the soil microbiome. Mycorrhizal fungi are only alive when the plant is alive, and then sporulate when the plant dies, which means they go down in the fall, and saprophytic go up in the fall when there is a lot of organic matter to digest. Saprophytic fungi break down dead organic matter and recycle it. This dirty pants experiment buries a pair of cotton undies for several months and watches as the fungi devour them. A high level of saprophytic fungi tells you that there is plenty of food for the fungi to release to feed the soil microbiome. Some of the most interesting mycorrhizal fungi, those fungi associated with the roots and the rhizosphere, are the arbuscular mycorrhizal fungi, which colonize 80% of all crops. And these are of great interest to growers because pairing the right AMF with your seed can have great benefits. AMF are purchased as spores, and there are many kinds of AMF and many different AMF spores. These spores germinate when they communicate with a nearby plant and must colonize that plant within 24 hours or die. Here you see the propagule form of the AMF spore in gray, and it secretes messages to the plant shown in red, and the plant replies with its own messages shown in green, thereby starting a symbiotic relationship. This symbiosis is mutually beneficial. Mycorrhizal fungi feed on carbon produced and released by the plant, and AMF in turn benefit the plants through multiple mechanisms. Some of these mechanisms are disease and pest resistance, as well as increased growth due to better nutrient access. The orange threads are the hyphae of the AMF, which increase the surface area of the roots. The AMF colonized plant gets more nutrients, especially phosphorus, as is indicated by the blue peas. Not only does AMF transport phosphorus, it extracts it from mineral components of soil by secreting chemicals that dissolve phosphorus. With AMF, the plant grows faster, and it is protected from disease due to stimulation by the AMF with hormones like jasmonate and ethylene to turn on defense genes in the plant that provide systemic resistance to infection. This systemic resistance protects the plant from viral infection as well as damage by pests. One of the advantages of AMF is drought resistance. Here are two plants, both deprived of water for 48 hours, but the one on the left, colonized with AMF, is doing much better. The one on the right here was colonized with AMF, and you can see how it is much larger and healthier than the one on the left. AMF offered disease protection. On the left is a control, and on the right is a tomato plant infected with a pathogen, but in the middle, the AMF shows that you protect the plant from the pathogen. If you are a grower hoping to increase yield and plant disease resistance by purchasing and using AMF spores or coated seeds, how will you know if the AMF you bought is actually colonizing the plant roots? Is it the correct species for my crop and my soil? Did I use enough AMF? How much AMF do I actually need? And is the expensive AMF coated seed I bought actually colonized? This is where microbiometer comes in and can help you figure this out rapidly and in field. To measure AMF colonization of roots, you're going to collect a root or a core sod, and you're going to shake off the excess dirt so that you get to the rhizosphere. And then once you're down to the rhizosphere, you shake that dirt and collect it, and you're going to sample and measure that with the microbiometer. What does the fungal to bacterial ratio in your forest or pasture tell you? Well, forests have a much higher fungal to bacterial ratio than croplands, which outside the rhizosphere area usually have a 1-to-1 ratio. And research indicates that with a higher fungal to bacterial ratio, you are sequestering more carbon. We also know that different fertilizers can greatly affect the fungal to bacterial ratio, which is decreased with nitrogen fertilizer, but responds rapidly to management changes.